First, a quick reminder of what we've discussed in the last session. We discussed 4 different types of passive membrane transport. We did agree on very fundamental concepts. And if you remember, generally speaking, anything would be passively transported from its high concentration to its low concentration. And we use the term down the concentration gradient, means from high to low. And this is totally passive. It doesn't require me to expend any energy or to consume any ATP in order for this membrane transport to take place. If something is passing down the concentration gradient, it doesn't require any energy to drive this movement. We called the transportation of a solute passively down its concentration gradient is a diffusion. And remember, diffusion is going to be different according to the chemical properties of the compound. So diffusion of a lipid-soluble substance across the membrane, which doesn't require the presence of a channel or a carrier, it's called simple diffusion. Compared to something like the sodium, for example, it cannot pass through the phospholipid bilayer because it's polarized, it has charge. And the phospholipid bilayer contains fatty acid tails, which would prevent the sodium from passing through it by simple diffusion. So it requires the presence of channels. So we call this as a channel-mediated facilitated diffusion. We also have seen another type of diffusion in which I'm transporting larger chemical compound, larger than sodium, but still I am not lipid soluble, so it still can't pass through the phospholipid bilayer. So this required me to have a carrier. So we call the transport like of glucose or amino acids across the membrane. By diffusion, it's called a carrier mediated facilitated diffusion. We'll see also another type of passive transport in which I am not able to transport the sodium or the solute, I'm sorry. The solute on one side of the membrane is too high compared to the other side. I can't transport the solute, but I can transport the water, which is a solvent in which this solute is kept. Remember, we are looking at concentration, not amount. So having too high solute content in a concentration means for me that I have less water. Compared to having less solute means I have higher water content. So I cannot allow the transport of the solute here across the membrane. This membrane is impermeable for the solute, but it's permeable for the water. So the water can travel. So where the water will be traveling from, it's going to be traveling from its high concentration to its low concentration. In other words, the water is going to be traveling from the low solute concentration which is actually high water to the side with high solute concentration. In other words, low water. So I'm traveling from low solute to high solute. And this is what gets many of you confused. They think because I'm traveling from low to high means I am moving against the concentration gradient and it's going to be a passive transport. No, it's not. Why? Why it's not a passive transport? Because the water itself travels from high to low, not low to high. But why did you say low to high? Because it's low solute, in other words, high water. And I'm traveling to the side with high solute, in other words, low water. But in terms of water, water travels down the concentration gradient, its concentration gradient. So is this passive or active? It's still passive. It is still passive, although you say the water travels from low solute concentration to high solute concentration. Please pay attention at this point. Very, very, very important. So we did agree that the transport of the solute down the concentration gradient is a passive transport. We called it a diffusion. Here, what do we call the transport of water from low solute concentration to high solute concentration? It's osmosis. Osmosis. We'll see important examples here dealing with the cells in the body. Remember, when you are surrounding the cells, would you surround the cells with fluid that has higher solid content than the cells, lower solid content than the cells, or the same solid content like the cells in terms of osmolarity? So outside will have high osmolarity compared to inside, or lower osmolarity compared to inside, or the same osmolarity like inside of the cell. Remember, if I did have higher osmolarity outside of the cell, what happens? The water leaves the cell and the cells will shrink. We call this solution in which I have higher solute content outside of the cells. This is a hypertonic solution. Hypertonic, extra solutes. Compared to if I do have less solute outside of the cells in the extracellular fluid, this will drive the water to move to the inside of the cell, as you see on here. Water is going to be moving from low solute to high solute. So water going to be moving into the cells, and the cells as a result will be swelling. So what we call this here, this solution, it's a hypotonic solution. So which one would you like to have in your body? A hypotonic solution or a hypertonic solution? Do you remember? Which one did we have in the body? Hypotonic or hypertonic? Hello? You got tricked again. Neither. Remember? Neither one. You don't want to have cells shrinking. You don't want to have cells swelling. You want the cells to remain in their regular shape. So which one would we have inside our body's extracellular fluid? It's an isotonic, neither hypertonic or hypotonic. Do you remember this? Do you remember this from last session? All right. So this was a quick reminder of the passive membrane transport, and our very first question today for the in-class activity is going to be, during osmosis, blank moves across the semi-permeable membrane to the side with higher blank. So in osmosis, what moves? What moves in osmosis? What moves in osmosis? Osmosis is a movement of water to the side with higher what? Too high what? Solute. Exactly. So the good answer here is going to be D, which is the movement of water to the side with higher solute. Moving on to the membrane, active membrane transports, active processes. We have two types of active processes. Both of them will require ATP, and ATP is the currency by which you're going to be paying for energy to move the solutes across the living membrane. It might be an ion, it might be sugar, it might be an amino acid. So we have many, many examples in which we're going to be using active membrane transport to transport a certain solute from one side of the cell membrane to the other. The active membrane transport, the active transport requires the presence of a carrier protein. And this will allow the movement of the solutes against the concentration gradient. In other words, it allows the solute to move from its low concentration gradient to its high concentration gradient against the concentration gradient. It is the opposite of what we've seen in the diffusion. So the example on here of the active membrane transport, it's one of many. So this is my cell like this. Something that you need to know is that outside of the cell, you have much higher sodium concentrations. You have high sodium outside of your cells, and inside of your cells, you have a high potassium. So I have outside of the cells high sodium, inside of the cells I have high potassium. So we're looking on here at this cell membrane. This is part of the cell membrane on here. I have lots of sodium outside of the cell and lots of potassium inside of the cell. So let me use another color here for potassium. But I still have some sodium inside and some potassium outside. I want to move this potassium from outside of the cell to the inside of the cell. And I want to move this sodium from inside of the cell to the outside of the cell. And we're going to learn later on in the nervous system and the muscular system, why would you like to do this? All right, so you see on here, the sodium is going to be trying to move from low sodium concentration to the side with higher sodium. And the same for the potassium. Potassium is going to be trying to move from outside of the cell, in which I have low potassium, to the inside of the cells, which has high potassium concentration. So each of them is going to be trying to move against its concentration gradient. All right, so we're looking here at a carrier. This is how it looks like. This is the inside of the membrane. This is my intracellular fluid. And this is my extracellular fluid. So I have a carrier. We call this carrier is the sodium, potassium, ATPase pump. So again, where is the sodium? Where do I have high sodium? Outside of the cell. And I have small amounts of sodium inside of the cells that I want to move out. On the other hand, I have small amounts of potassium outside of the cell. I want to move them inside the cell where I have a high potassium content. So what I will do, this is my ATPase. It's going to be binding to 3 sodium. So I've got 3 binding sites for sodium. So the carrier is facing the inside of the cell membrane. It binds to 3 sodium. Once it binds to the 3 sodium, it will be getting its energy by hydrolyzing an ATP into an ADP and an inorganic phosphate, a phosphate group that would be kept attached to the carrier. So this is how the carrier is going to look like. Those chemical modifications will change the shape of my carrier. So the carrier, rather than being facing the inside of the cell membrane, it's going to now rotate and will be facing the outside of the cell membrane. So where is the sodium now? If you see, the sodium is still attached, but now the sodium did move from inside to the outside. And this is a phosphate. So what happens when the carrier is now facing the outside of the cell membrane? The sodium gets released. So it will leave the carrier. The same for phosphate group. And the carrier now will expose two binding sites for the potassium. So now two potassium will bind to their binding site. And again, the extracellular site. So once the two potassium bind to the sodium potassium ATPase pump, the ATPase pump rotates another time to face the inside of the cell. Moving the two potassium to the inside of the cell membrane. Those two potassium will be released to the inside of the cell, and the carrier is going to be ready for another cycle. It's going to be binding to 3 sodium, will be binding to an ATP that will be releasing the energy needed for it to rotate and move the 3 sodium to the outside, as you see on here. And then once it releases the 3 sodium to the outside of the cell, what happens again? 2 binding sites for potassium will be exposed, and this will allow the carrier to bind to the 2 potassium, and it will rotate again, moving the inside of the cell, and this will allow it to release the potassium and again to expose the 3 binding sites for the 3 sodium. So it binds to other 3 sodium, binds to ATP. And once it binds to ATP and hydrolyze it to release the energy, it's gonna be modifying its shape, rotating to face outwards, releasing the 3 sodium and the phosphate. It will expose 2 binding sites for 2 potassium. 2 potassium will bind, the carrier here rotates another time facing the inside of the cell membrane, and this will allow it to release the potassium and start a new cycle by exposing 3 binding sites for sodium, binding to 3 sodium, and hydrolyzing an ATP will allow it to rotate, moving the 3 sodium from inside of the cell to the outside of the cell. Once the 3 sodium gets released, this will allow the carrier to expose 2 binding sites for potassium. And this will, once the 2 potassium bind to the carrier, this will allow the carrier to rotate to face, again, the inside of the cell membrane, and will be releasing the 2 potassium to the inside of the cell and will be ready for a new cycle. And the cycle keeps going all the time in the background. the sodium potassium ATPase pump is going to be activated all the time in the background. It's always going to be trying to move the sodium from inside of the cell to the outside of the cell, and it's trying to move the potassium from outside of the cell to the inside of the cell. And this is what keeps the concentration of potassium high inside your cells and the concentration of sodium high outside of your cells. All right, it's very, very, very important for you to be able to conduct electrical signals through your muscle cells and nerve cells. It's very, very, very important. Any questions? Any questions concerning the sodium potassium ATPase pump? So is this an active or passive? Can you remind me? I am moving the sodium from low to high and the potassium from low to high. Does it require energy to do this? Did I consume any energy? Did I use ATP? Yes, so this is an active or a passive transport. It's an active membrane transport. All right, so another type of active membrane transport is the vesicular transport. And vesicular transport is going to be classified according to whether I am moving something in or I'm moving something to the outside of the cell. The vesicular transport is going to be mainly concerned with the transport of large particles and macromolecules and fluid across the plasma membrane. This requires ATP, requires energy. That's why we consider it as an example of active membrane transport. If I'm moving inside, something from outside to the inside of the cell, it's called endocytosis. To the inside of the cell, cyto is cell, endo is inside. So if I'm moving something from outside in, it's endocytosis. If I am engulfing a solid particle like a bacteria, for example. So I've got a bacteria on here. All right. I have a types of immune cells that can engulf the bacteria and break it down. All right, so what will it do? It will extend cytoplasmic processes here, extensions of its cell membrane. To surround this bacteria. And those two sides of the cell membrane will be fusing together. So now where is the bacteria? The bacteria is inside a vesicle inside my cell right now. See, this is the vesicle. And it's now inside my cell. So I did ingest a solid particle. So if I'm ingesting solid particle, it's called phago. Phago is to eat. Cytosis. All right. Example of cells that have the ability to perform phagocytosis, to engulf solids. It's gonna be my big eaters, macrophages. Macro means large, and phago is to eat. So macrophages, those are my big eaters. This is a type of white blood cells that have the ability to engulf the pathogens. Other cells will need endocytosis, will be performing endocytosis, but this time they will be uptaking fluids. Like, for example, I have fluid, and this fluid is gonna be containing fat in the small intestine, let's say, the small intestine itself, like this. Or whatever kind of nutrients that you want to have to the inside of the cell, and you don't actually have the ability to get it in unless you take it in a liquid form. So what the cell will do on here, it will come the same way. It's going to be forming arms to surround the fluid-based substance. And those two sides will be fusing together. And what I will end up with, a cell. And inside the cell, I have the vesicle containing the fluid and the solute I wanted to move in. So here, if I am moving fluids or fluid phase endocytosis from outside to the inside of the cell, it's like the cell is drinking. So we call this is pinocytosis. So if I move in a fluid-based substance to the inside is endocytosis. We've got two examples of endocytosis. If you are moving solids, like engulfing a bacterial cell here, it's going to be phagocytosis, eating. If I am moving a liquid-based substance, a fluid-based substance to the inside of the cell through vesicular transport, it's a type of endocytosis, but it's like drinking, pinocytosis. What if I am a cell that will be releasing hormones? So I have vesicles that will be containing hormones. Those are my hormones that I want to release. But those hormones are large chemical structures, so I can't just release them to the outside through a channel. They are too big. So what will I do? I store them in vesicles, and those vesicles will be moving towards my cell membrane, and they will fuse with the cell membrane. They will become part of the cell membrane. And as they fuse, now the content of the vesicle will be released to the outside of the cell. So now the hormones I wanted to transport are moved from the vesicles to the outside. If I am a hormone, it's going to be conducted to circulate within the blood. All right, so what do we call this vesicular transport, in which I'm transporting something from inside of the cell to the outside? Outside of the cell is exocytosis. Exocytosis. So I got a question here. Do the walls of the vesicles have the same properties as the cell membrane? Yes, it's the same chemical structure. So it's phospholipid bilayer. You see on here, phospholipid bilayer. It contains the hormone or whatever you want to release outside of the cell. Those vesicles will be binding with the cell membrane, will fuse with the cell membrane at special sites of receptors. And you see on here, fusing with the cell membrane will allow the vesicular contents to be released to the outside of the cell membrane. The same exact concept here. I'm taking part of the cell membrane to surround the bacteria, and as the cell membrane folds together, this will allow the vesicle, which is part of the membrane, to dislodge from the rest of this cytoplasmic membrane and to move to the inside of the cell. So yes, the vesicle has the same chemical structure and properties as the cell membrane. All right, we can have a 10 minutes break, and we will come to discuss after break the cytoplasm and the cytoplasmic organelles.